Today, we are gonna discuss about one mechanism of action of antibiotics, which is interfering with DNA and RNA synthesis. Like any other organism, bacteria must convert their genetic information into functional molecules in order to carry out their life processes. This is done by using DNA as a blueprint to synthesize RNA, and the production of proteins is done through the translation of RNA. But what will happen if these processes are disrupted? You'll soon find out. There are three antimicrobial agents that target nucleic acids, which are DNA and RNA. These are metronidazole, quinolones, and rifamycins. Wouldn't it be toxic to our body cells? Well, bacterial enzymes that carry out the synthesis of DNA and RNA appear to be different from ours, thus enabling the production of toxic drugs such as these three. First, metronidazole. It is a prodrug that requires reductive activation of its nitro group. Upon diffusing across the cell membrane of the bacteria, it is then converted by the bacterial enzyme called pyruvate ferredoxin oxidoreductase or P4 into nitroradical anion. Nitroradical anion is a toxic agent that causes strand breaks and mutations in DNA and eventually lead to bacterial cell death. Next are quinolones. Involves interaction with topoisomerases which are responsible for the unwinding and unlinking of the DNA. There are two principal topoisomerases which are DNA gyrase and topoisomerase 4. DNA gyrase is for the unwinding and relaxation of supercoiled DNA, while topoisomerase 4 facilitates the separation of the daughter DNA molecules after replication is complete. Quinolones bind to DNA gyrase and topoisomerase 4, which inhibits their function, prohibiting the DNA synthesis and cell growth, leading to bacterial cell destruction. Lastly are rifamycins. They bind with RNA polymerase, which abrupt or stop the synthesis of RNA, prohibiting the production of proteins that are essential for the survival of the bacteria. And that concludes our discussion.